Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension here at UW-Madison. And on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to welcome back to Wednesday Night at the Lab, Prof Burkard. He's with the School of Medicine and Public Health and the Department of Medicine. He's also with the Carbone Cancer Center. He was born in Buffalo, New York, and went to high school there at Williamson North High School. And then he went to SUNY Buffalo and studied mathematical physics. And as I said to him, is there any other type? Then he went to the University of Rochester to get both his MD and his PhD in chemistry. And then he became a resident at Cornell Weill Hospital in New York City, and then moved over to Memorial Sloan Kettering for an oncology fellowship, in New York City. And then he came here in 2008 to UW-Madison. And tonight he's going to talk with us something pretty near and dear to my heart, because I went through this personally with my dad about 10 years ago. The whole issue of somebody that you know, love, gets a cancer and they can't seem to be quite able to figure out what it is. And I think if this were to happen now instead of 10 years ago, things might have worked out a little bit different. Tonight, Mark's going to talk with us about transforming cancer treatment with precision medicine. Please join me in welcoming Prof Burkard back to Wednesday Night at the Lab.
Thank you, Tom, and thank you all for coming out tonight. It's a pleasure to be here tonight and to talk about some of the things we're doing in precision oncology and how we're trying to embody the Wisconsin idea by reaching out to the state and doing so. So, the fundamental basis of what I'm going to talk about today is this: how do we classify cancer? And as a medical oncologist, we differentiate ourselves and our patients primarily by over 100-year-old idea, which is we classify the cancer by where it started from in the body. And that is very important, especially if you're thinking about surgery. So if you're a surgeon, how you operate on the lung or the colon or the breast is very different. And so when you're classifying it by what type of surgery you're going to do, it's very important to classify it by where it started in the body. But that was a very old idea that, harkens back to the 19th century. I mean, we had breast cancer going back hundreds of years, and lung cancer and colon cancer. We knew about these diseases, and surgeons have been operating on them since the dawn of anesthesia, and sometimes, sadly, before. But we also now know that we can do much more with medicines for these cancers. And so it's now all of it suddenly becoming more and more important to classify these cancers not just by what organ they started in, but by the genetics. What are the genes driving the growth of each individual cancer? And as I'm going to show you tonight, it's highly variable what genes you find in an individual person's cancer. So let's talk about a case. So I think about patients. I had clinic today. This wasn't my patient, but it's something like what many of us oncologists see in clinic. So imagine we have Ms. Jones. She's a 67-year-old. She had blood in her stools. It turned out, unfortunately, that this was colon cancer, and she was treated with surgery. So while we hope that many of our patients do well, unfortunately, as with many of our patients, a couple years later, something else happened. A blood test was abnormal. They found a tumor in her liver and biopsied it and showed that this was the colon cancer back. So this is where patients many times come to the almost exclusive care of the medical oncologists, and we're faced with, what do we do? And if I look at any guideline, what we do is we start to classify her cancer. So if we classify it by colon cancer, you have metastatic colon cancer. I go to the guidelines and I have a list of drugs that are approved. And there's standard chemotherapies that have been around for, in some cases, quite a while. such as 5-fluorouracil, discovered by Charlie Heidelberger here at the University of Wisconsin. So shout out to UW there. But we also know that if we look and we start to uncover the DNA inside Ms. Jones' tumor, we may find a specific set of driving genes specific to her cancer that's not necessarily common to all colon cancer. And sometimes, as I'll show you, those genes may be more common in a completely different type of breast cancer, such as breast cancer, but found in Ms. Jones' colon. So the idea here is if we can actually peer inside each patient's cancer, maybe we can be smarter about choosing the therapy. Maybe we don't simply go to the guideline for colon cancer, but we look for what is useful for her. So I have to back up here because often I get contacted by people after these talks that, I have a friend or a colleague who has stage 2 breast cancer. Should we do precision medicine here? And what I want to say is for the routine use in the clinic today, this is being used first in people with metastatic incurable cancer, because a lot of this is done under the research. And if we have a patient with early stage breast cancer or even advanced cancer that can be cured with a standard approach, we don't really want to skip that. And we only want to embrace this approach in the setting of very careful clinical trials that show we help people without hurting them. But in patients, and we have many with metastatic cancer, this is something we seek to implement today, as I'm going to show you. Also, what I should tell you is tomorrow we hope to integrate this in early stages. We learn more about our precision approaches and precision therapy. We are moving these into early stage cancer, but in a very stepwise approach. So when I talk to patients and families, I have to be very careful about talking about DNA tests. And when we're dealing with someone with cancer, we're dealing with basically two sets of genes. One is from the person, called germline, and one is from the cancer, called somatic. So what do I mean by that? So the germline is the genes in all your cells in your body that you inherited from your parents. And usually we test that by taking blood. You can take saliva or anything else, but all of our cells in our body have those genes. So you could learn by testing those genes what you've inherited from your parents. You could find out, do I have a gene I inherited from mom or dad that predisposes me to a cancer or another disease? And you can use that primarily, the major use is to find out that, gee, I have a risk of prostate cancer, maybe I should have screening more, for instance. So that's germline, but what I'm talking about today mostly is somatic. What are the genes driving the unique growth of the tumor? And it turns out our tumors, when we get cancer, are us. They have everything we inherited from our parents, but then some more changes have occurred to drive the growth of those cells in an abnormal way. So today what I'm talking about is the somatic mutations, or the mutations that are driving the growth of the tumor that may not be in the rest of the person. I might not find that very well in blood, but I can do a test of the tumor. And I can find what genes are driving the growth of each patient's tumor. And by doing that, I can find potentially ways to turn off the growth of the tumor with medicines that turn off the enzymes that those genes encode. So that's what I'm talking about today. I'm really talking about these somatic mutations in patients' tumors that often are very different from what they inherited from parents. So why are we talking about this today? We've known cancer is a genetic disease for decades. Okay, so it's really no mystery. But why are we just starting to, get smart enough to look inside each patient's tumor to find what drives it? And basically the driver is technology. So technology has changed such that we can now rapidly sequence people's genes and their tumors. And if you recall, I don't know how many of you recall this, but in 2001, of course, there was a great announcement. We had the Human Genome Project completed. It cost $100 million, took over 10 years. It used basically an updated version of the 1978 Sanger sequencing technology, but to do it on a massive scale. So what the NIH did next in 2001 is invest in technology, in developing new tools and sequencing technology that could do this at a massive scale, such that now what cost a great, tens of thousands of dollars in 2001 cost pennies for the same amount of DNA sequencing information. So what this is saying, this is a megabase. So if I want 1 million base pairs of your DNA sequenced, I can do that for under 10 cents now. So this is why we're getting to this today. So how do we do it? Well, usually what we do is we work with our friends in pathology. Our tumors under the microscope might look like this, for example, where we see a lot of cells, sheets of tumor cells that have grown from the biopsy. We take and extract the DNA from that. We can place it on these chips. Often different technologies are used. This is an Illumina chip, which is one of these massive sequencing technologies that can sequence a lot of DNA at low cost. There is technology that uses DNA bridges and copying using an enzymatic approach, and you get millions of reads of DNA. So we could do that with our patients' tumors and look inside each patient's tumor to find what's driving its growth. So we find the genes that drive the growth of the tumor, and then we have to use that to try to help our patient. And this is where it can be challenging for many reasons. But one reason is, it's hard for oncologists to keep on track of the new drugs for colon cancer and breast cancer and everything else. But now you add this whole other dimension of, now you have to learn about all the genes that are driving the growth of cancer and what you can do about it. So as part of what we like to do at the University of Wisconsin, which is serve the people of the state of Wisconsin, we set up what we call the Precision Medicine Molecular Tumor Board. And we got all the relevant experts from UW-Madison and from collaborating oncology practices around the state. And we set up a web conference. And what we allow is any physician who does this test for any patient anywhere in Wisconsin, free of charge, can submit the test result to the tumor board, has the option of calling in through the web conference, and get a recommendation of what you can do with that information. So we look at the DNA sequence, we try to figure out if the mutations we're seeing are actually just there or are actually driving the growth of the cancer. If they're driving the growth of the cancer, we look at whether there are medicines that can be used to turn those enzymes off. And sometimes, but not always, we do find things. So we send to each physician a recommendation letter. So everyone gets a letter. It tells them whether there's a clinical trial of a drug that can be specific for the patient's tumor, whether there is another treatment, or in many cases, frankly, we don't find a treatment. So there are many genes that we know drive the growth of cancer that we don't have drugs for that are very challenging drug targets, some of which because they're turned off. So, we have two kinds of cancer genes. We have those that are turned on, those are oncogenes. They get mutated, they turned on, and the enzyme is high. So you can turn them off with a drug. That works pretty well. But the other set of genes are things that get broken and turned off when they're mutated. And when those get mutated, there's really not a drug to turn them back on. So this set of genes is much harder to target. So we find both kinds in all our patients almost, but in a small fraction of patients, we have a very good drug. To show you how complicated this gets, I just want to unveil what it looks like. So this is a picture from a paper we published a couple years ago. This was the first 30 or 40 patients who were part of our tumor board. And each row is a different patient whose tumor was analyzed by DNA sequence. Each column is a different gene that's mutated. And what you see is just about every row is different. Every patient's tumor has a different set of mutations. I would challenge you to find 2 rows here that are identical. And what you also see is, although some genes are common, the one at the far left is called TP53, or the classic P53 tumor suppressor. That's one of those genes it's broken off, so we can't turn it on. but we can find some other genes less commonly mutated in patients. And at the far right, you see there's about half a dozen patients of this whole group that we were able to find a drug. So we realized pretty quickly that we were only going to help a small fraction of patients, but if we helped them a lot, it might be worthwhile. So, but we wanted to challenge ourselves to help more patients. So we didn't want to just help 7 of 35 patients. And we knew what we needed to do is not just have the standard FDA-approved drugs, but work with researchers and pharmaceutical companies all across the world to bring the latest drugs, the best drugs that are for specific genes into the clinic. So we had this idea that maybe we could help a lot more of our patients if we did this. If we set up a system where we could test as many patients as possible, and maybe we would find the patients that would match a drug. And this was a hard thing because if you look at the bottom, people call these basket trials or baskets, where you have genes like NTRK or ERB2 or FGFR that are mutated maybe in a tiny fraction of patients, 2%, 1 to 2%. If you have enough of these baskets, it adds up and you help a lot of patients. But each basket in each trial is only going to help a small fraction of patients. So what we realized pretty quickly is if we did this testing for as many patients as possible, and we're screening thousands of patients for these tests, then we could work with these companies and get the best drugs for our patients. For that 1% for this, for 1% for that. If we couldn't do this widely, if there were too many barriers and we were only testing, say, 50 or 100 patients a year, this whole process would fail. This would fail because we'd only get 1 MET a year. And so we would never be able to bring that trial to Wisconsin. So this depended on broad implementation of testing to help as many patients as possible. Now, so we realized we were kind of in this catch-22, this chicken and egg problem, where we could help more patients with the latest drugs that are being developed, but we could only do that if we were testing as many patients as possible. So we would go to the pharmaceutical companies and we would say, would you do this? Are you interested in working with us? And they'd say, well, how many patients are you checking? If you're checking enough, it'd be worthwhile. And then we go to the doctors and we say, how come you're not doing this testing on all your patients? And there were a number of barriers, but they're like, well, a lot of times I do these tests and I can't do anything to help my patient, so why should I do it? So it created this chicken and egg problem that we felt like we had to break down the barriers to doing this testing to make it easy for physicians. And we identified a lot of barriers. So over the years, the routine in clinic was just that when the doctor decided to do a test, they would try to run it. And the doctor and the nurse would do a lot of paperwork. Maybe the patient would get a big bill for running the test. And we discovered that there were 4 main barriers that we needed to break down to make this routine. And so we set out to break down each one. The first one was that the clinic flow. So, I have a clinic with about 40 partners and maybe 50 nurses. And so when the doctor wanted to run a test, the doctor would turn to a very busy nurse who's getting phone calls and barrages of things from other patients and saying, I want to do this test, let's fill out the paperwork. That nurse would find the 5 nurses in clinic who are familiar with that, where they would try to run that test, and then it would get run. And that one patient would, and the doctor would know about it, and it wouldn't be shared across the group. So they'd only be known to them. They wouldn't know what anyone else is doing. So what we decided to do is centralize it. And in the late 2017, with the support of the Cancer Center director, we were able to hire staff, first Kayla Lemon and then Hannah Hautler, who were able to help overcome this. And I very distinctly remember my meeting with the nurses, because I met with all the nurses and I said, you aren't going to need to do this anymore. We're going to do it for you. And they looked at me dumbfounded because they'd never been asked to do one thing less. And actually, they didn't believe me. what they said was, okay, what's the catch? What are you really asking here? But what we did by taking that on and saying, we're going to take all this paperwork away from you and just do it. All you need to do is let us know. And once we did that, we knew every patient was being tested. We could centralize the process. We could coordinate with our clinical trials. And we had over 20 patients a month that we could test. But we also, as we were going through the process, we noticed other problems. So we broke down that barrier by giving away some help. The next problem was dealing with payers. So the way insurance companies work is they don't really support doing a lot of tests on patients, and maybe they shouldn't until that test has been proven to improve the outcome of enough patients. And so what we're doing today is we're helping small fractions of patients profoundly, as I'll show you, but we're not helping large fractions of patients until we get these drugs developed. And so as an academic center, we said, we can't deal with this. We can't be charging patients hundreds and thousands of dollars. Who's going to want to have this test? And yet, if we don't do the test, we can't have this program. And insurers weren't, ponying up. And as you know, when in medicine, we deal with many different payers. So even if this insurer will cover it, we're dealing with dozens of these companies. So that was a huge time sink trying to organize and work through that. So what we decided to do, we had the opportunity to work with a company out of Ann Arbor, Michigan. So these are not badgers. They're wolverines, but they were quite helpful. And Scott Tomlins and Dan Rhodes set up this company. And their idea was, the problem with doing this testing and developing the drugs is we can't do the testing because the insurance companies don't pay for the testing because we haven't proven the testing's useful yet. And so what we have to do is get the drug companies to pay for the testing. And we'll make them pay for the testing so they can develop their drugs. So they set up this network across the country. We implemented this widely, both at UW Carbone Cancer Center, and got them integrated with many partners around the state. And so they pay for the testing for our patients. And the catch for us is we are expected to, when we find a match for one of these rare patient populations, to let the patient know there's a clinical trial available and do the work to bring it here to Wisconsin. So it created this win-win situation where we didn't have to deal with payers. So we broke down that barrier and were able to test everyone without charging patients hundreds of dollars or dealing with insurers, which was a nightmare. So the next thing we ran into was this. So pretty soon we were calling the pathology lab multiple times a day and say, I need you to go find this biopsy sample from this patient from March 25th. And the pathology department, what they do is they take all the biopsies and surgical specimens, they put them in paraffin-embedded, formalin-fixed blocks, and there's a huge library there. And so this library goes back at least 10 years. They're, by national guidelines, required to store these 10 years, and ours goes back further. And so we had all these librarians, so to speak, running around getting these blocks from dozens of patients down in pathology lab. And pretty soon they got upset at us because it was eating up all their time. And they're like, well, we have patients who are going in surgery now. We have to do we have to take care of them. So we said, okay, we have to get over this. So this is some of my wonderful colleagues in pathology, Christina and Daria and William and Stephanie. And they worked with us, and we were able to provide some money to have another librarian. So we have our special librarian that we pay for that goes through the libraries like that, those drawers of blocks that you see down there. So, and not only that, but Daria and Christina and Stephanie have to look at each slide before we send it out and make sure it actually has adequate tumor to do DNA testing. So they've been wonderful colleagues making this work. So we were able to break down that barrier. So, and then finally, the last problem was, okay, we're going to find now we got everything working. We're going to screen hundreds of patients a year, maybe get over 1,000 at UW. And now we have to bring the clinical trials. But all these trials are for, a small fraction of patients. So which one do we bring first? We can't open a dozen clinical trials all at once because they take a lot of work for each one. So what we did was, first of all, I got a wonderful team, my colleague Natalia Uboa and Jen Collins, who's the manager. We created a new research group in the Cancer Center called the Basket Group to open these trials. And we challenged ourselves to reimagine clinical trials. So usually when you run a clinical trial, there's a lot of steps to get started. So usually we work with a sponsor, often a pharmaceutical company that has developed a drug. We do contracting, budgeting, we do a scientific review called PRMC. We see whether it's compliant with insurers and payers. And then we go through the institutional review, the ethics review board. They review it, then it goes to a national ethical review board called the Central IRB or CIRB. Then it goes to the pharmacy and they create all the order process. And then we activate the trial. So this takes 3 to 6 months for each one, typically. And then we start waiting for the patient to show up. So the patient may or may not show up, but if, in these rare patient populations, if we're not screening enough, it may take months to find that first person who needs that. So we said, this just isn't going to work. We can't do this with a dozen trials and then wait for the patient to show up. So what we did was we reimagined it. We met with each group among those and we said, what if we do this? What if instead of having the patient at the end, we do the minimum amount of upfront work, and then as soon as that patient shows up, we want you to commit to doing all these reviews as quickly as possible in time to help this patient, so that we can find the patient for this particular trial, and we can work with you, and we want you to get it done in less than 4 weeks, so we can treat that patient and help them. And with their help, we were able to do that for the first time. It was the fastest trial opening we had. Was one of the trials we did last year. Took about 4 weeks from finding the patient to getting the first patient treated. And it turned out the research nurses loved it because usually they get trained on a research protocol and then a patient may or may not show up 6 months later than when they might or might not remember their training. But with this process, they were trained and then the patient was right there. So they used the training right away. So that broke down that barrier. So we felt like we were finally in a position where we could get through this chicken and egg problem by breaking down all these barriers to start doing testing on all our patients without charging them and bringing the drugs that they need for each different gene. So again, the challenge of precision oncology is there's so much diversity in tumors, you have to do a lot of work. There's not one solution. You have dozens of solutions, and you have to be prepared to deliver all of them at once. So that's how we set up that program. By doing this, we were able to serve patients from all over the state. So this is where patients came to our cancer center to be tested. So these are the patients that came to UW Cancer Center and then had the testing done over the past year. We've had many other patients test at our collaborating sites that aren't included here. But as you see, this serves pretty widely the region. And if you look at who was tested over the past year, it's, again, a very diverse set of cancers. So we've tested a lot of colon cancer, lung cancers, breast cancers, but the whole battery of the solid cancers that can occur in patients have been tested. So we're serving a lot of diverse clinics and patients. We also thought that it's great to do this at UW, but as the state university and with the Wisconsin idea, we wanted to reach other patients. And we didn't want to tell patients they have to come to UW-Madison because although we love to see patients, we probably couldn't see everyone. Because it's a hardship on patients. So we wanted to work with their oncologists in their community to deliver the same level of precision oncology. So we got these collaborators from around the state, Ben Parsons in Gunderson, Ruth Warren at Green Bay Oncology, Mike Thompson in Aurora, Matthias Weiss in ThetaCare, and Timothy Goggins at Fox Valley, and we've recruited others since then. So we could reach out to the entire state. And we said, for free, you can submit these cases to our tumor board, and we'll connect you with the Strata Oncology if you want to work with them to get free testing for your patients as well. So this has rapidly taken off. Last year was when we finally broke down all those barriers, and through our tumor board, we reached almost 1,000 patients coming through, getting a recommendation. That doesn't mean we have found a drug for all of them, but we certainly found new treatment options for many of them. And in 2019, we're on target to have well over 1,000 patients through this. And as you can see from the map, these patients come from all over the state and health systems all over the state. So what does this mean? Can I give you an example of some patients who came through this process? Well, first, let's reimagine Ms. Jones. We talked about Ms. Jones, so she had this fancy DNA testing of her tumor, and maybe, for instance, for her, we would find in colon cancer a mutation in a gene called PIK3CA. So that test result would be submitted to our tumor board. If it happens at our cancer center, it's automatically submitted. Other cancer centers, they can submit free of charge through a secure portal. And we would look at what we could do for her. But how do we determine what treatment to recommend? What do we do there? What's happening in the tumor board? So I just want to unveil what we do a little bit. So how do we determine whether Ms. Jones' mutation matches a drug? And so, for example, one thing we can do, in her case, what we found is PI3 kinase mutation at E545. And so what this is a map of the protein, the PI3 kinase protein, the different domains on that protein that have known functions. And then this is called the lollipop diagram, and what it looks is publicly available data on all the mutations ever found in cancer on this gene. And what you can see is there are many mutations at the E545 position, at 545. So we can use that information to say, okay, since this is found really commonly in cancer, it's probably driving the growth of many of these cancers. We can also take that information and bring it back to the lab. So if it's E545 that's mutated to an A, for example, we can look at how that changes the behavior of this enzyme in a biochemical assay or a cell-based assay. And for example, this gene, PI3 kinase, is a well-known cancer gene, and there's even a crystal structure. So we can map what would happen to the crystal structure as right down in the bottom right corner, that amino acid is mutated. And we can see how that's driving the broken on switch, if you will, of that enzyme. So as I said, what we find, and one of the challenges of precision oncology is many drugs are only going to work in a tiny fraction of patients. But if you keep looking for enough patients, you find some that you can help a lot. So, for example, in breast cancer, we've found mutations in ERB2, also known as HER2, that we have drugs in clinical trials for. So we've enrolled a number of patients on treatment for that. We're working on clinical trials for patients who have estrogen-resistant breast cancers. That happens through mutation of the estrogen receptor encoded in the gene ESR1. So that's a common mechanism of resistance. We found those patients. We found a patient who was referred to us with breast cancer who had an EGFR exon 19 mutation. Now that's a mutation that occurs in about 5 to 10% of lung cancers and 0% of breast cancers. So we think her cancer was actually misclassified as a breast cancer, but in any case, we have very good drugs for turning off that enzyme, and so we were able to repurpose that. We found a breast cancer, and this is extremely rare, with a gene called RET, which is an enzyme another protein kinase for which we have very good drugs in clinical trials. So we were able to find drugs for that patient. There's another protein kinase we found amplified, which means it was, there were many copies in the genome of a breast cancer patient. And that's very unusual to find because usually we find that in lung cancer. And so, for example, this was a patient that my colleagues at Gunderson submitted to our tumor board. This is a patient who lived on a farm in western Wisconsin, and she unfortunately had this breast cancer. And as you can see here on the panel A, the one on your left, this is a PET scan. So what it shows is the glucose uptake. And at the top you see her brain, that should take up glucose. And at the bottom you see her bladder, that's where the glucose gets excreted when it's injected, so that's normal. But all in the middle, especially around her left lung, you can see there's a lot of white there. And so she had a lot of cancer wrapped around that left lung in the pleura. And because she had a MET amplified cancer, they gave her a drug that turns off the MET enzyme. And sure enough, about half a year later, what happened to her is shown at the right, where the cancer was diminished. You see a lot less white around her left lung on the left. And below, if you look very carefully, you can see that same thing in CAT scans. Normally, the lung looks black on a CAT scan because it's full of air. And in the far left, what you see is the left lung is full of fluid, and that goes away. That went away. So this was a patient that benefited from this. We would have never known to give this drug to a breast cancer patient without doing this. This is a very, very rare mutation. So one of the most interesting mutations are these genes called NTRK. So these are neurotrophin receptors that are usually found and expressed only in neurons. In a very tiny fraction of breast cancers, these genes are moved to a different spot in the genome by a mutation event. And then the new spot turns it on and causes it to be expressed as a protein and turns on an enzymatic activity that makes the tumor grow a lot. And when this happens, it causes cancer. But it's very rare. These are found in about 1 in 1,000 to 3 in 1,000 cancers. So you never find them unless you search a lot of patients. But when you find them, things like this happen. So this was a patient my colleague met who had a type of tumor called a Gi Stromal tumor, which is a rare type of sarcoma. And it may be hard to see under the light, but what you see is this is a CAT scan of her abdomen sliced through like this. And what you see is this big soccer ball shaped thing right in the middle. And that's her tumor. So this tumor was out of control in her abdomen. She was extremely ill from this by June of 2017, and it hadn't responded to any of the standard GI stromal tumors treatments. And this patient had a sample sent for DNA testing. And sure enough, this patient was one of the 1 in 1,000 who had the NTRK. And it turns out we have a very good drug for that. And so this patient, that tumor melted away. So the tumor, you can barely see it, is the size of a golf ball between the two pelvic bones in the middle. And what you see is a normal healthy liver and a normal abdomen. So within a few months, the tumor had basically dissolved. And so she went from being very close to dying and going to hospice, which was her condition in June of 2017, to be a person who takes a pill once a day and is healthy walking around like us, which is an amazing thing. And she's still doing well today, fortunately. So that's one of them that, when I talk to my colleagues around the country, and some of us ask, like me, why don't you test everyone? Because what if you're missing one of these? And they say, well, that's so rare. And so there's this unusual philosophy of, do we pay to test everyone? Do we test everyone? Do we do all that work to find one of these in 1,000? And my answer is yes. I think we should do that in Wisconsin, and so that's what we try to do. So another example of that is I had a patient, or my colleague Dusty Deming had a patient where he found this unusual type of colon cancer. So this is a set of CAT scans through in his liver on your left, and in panel A you can see there are like these holes. It looks like Swiss cheese in the liver. The liver is the organ on the far left. And this patient came to Dr. Deming, was an elderly gentleman, and he said, don't give me chemotherapy. I know that's standard for colon cancer, and maybe I'll live a little longer, but for me it's not worth it. So Dr. Deming said, okay, well, let me at least do, test the tumor as we do for everyone. And the patient had a gene that was amplified called HER2. Now, this is usually found in breast cancer. This is about 20% of breast cancers, a smaller fraction of esophageal cancers, and a very tiny fraction of colon cancers. Maybe a couple percent have this property. So because of that, he said, why don't we give you the breast cancer drug? We know this works in HER2 amplified cancer. You basically have breast cancer that started in the colon. So we're going to treat it like that. And sure enough, as you can see in the second set of panels, B, at follow-up, the tumor regressed remarkably. And by 3 months, the CEA, which is a measure of how active the cancer is, it's a protein that appears in the blood when cancer is active, it greatly diminished over time as we could see the tumor shrink. So this was another patient, skipped chemotherapy, had a great response. Now these are not the common patients. I'm not showing you the most common. I'm showing you some of the best success stories. So I don't want to oversell this and say we help everyone. But what we're trying to do is find another set of treatments and to search, uncover, turn over the stones for everyone so we can help as many patients as possible. So any of our patients, even if we don't find something for them, they can still have standard treatments. We didn't take that away. We didn't even do anything to them. We just took a sample of the tumor that we already had and analyzed the DNA. So how are we going to use this going forward? So what we envision is a setup where we start out with this statewide network. Our vision and purpose here is to get everyone tested so we can help as many patients as possible. So we want to break down all those barriers and help all our colleagues and help every patient, every cancer patient with incurable cancer around the state. We want to keep track of these patients. So for example, if we have a new drug, there's a new drug for a gene called KRAS that's very exciting. So we want to be able to identify all those patients that are out there when we get a new drug available. So we're tracking them. And then we want to use that information of who our patients are to do clinical research, to bring new drugs to clinic, and to help develop these drugs that are going to help these small fraction of patients by their genes. And then we want to work with our colleagues across the university to understand the biology. And so as we do these tests, we can annotate all the samples we have in the various banks that we have at the Cancer Center and use those banks to work with collaborators across the university to understand the biology and be more strategic about how we do this in the future. So what we hope to do with this is find options for all our patients. I estimate we have I don't know how many people get metastatic cancer in Wisconsin each year. What I do know is about 4,000 people die of cancer each year. So that number hasn't changed over the years. I think about 4,000 people in Wisconsin get metastatic cancer each year. And so what we'd like to see is all of them. We'd like to do this testing for all of them to help as many of them as possible. So this is something we're doing today, but we've also thought a lot about how we might help our patients in the future. And I want to introduce you to a really interesting patient I met, Peg, who's in the Wisconsin State Journal, so she's okay with me talking about her. As we're helping patients, how are we going to help patients tomorrow? We have to learn from our patients. So Peg is interesting because she had breast cancer in 1978, and it recurred and became metastatic in the early 80s. And she's alive today and doing well with metastatic breast cancer. Now, most people don't live 30 years with metastatic breast cancer, so there's something very unusual about her. So if you think about this, this is what we see with breast cancer. So of all the people with breast cancer who come into our clinic, fortunately, when we do surgery or treatment of the breast, most of them live. About 80% of them, we never see the cancer come back and they live. But about 20% of them, on your right, have recurrent metastatic cancer. And most of them, unfortunately, die of breast cancer. But once in a while, there's someone like Peg who lives with metastatic breast cancer. And so we thought we could learn things from her to understand why. Now, what happens with metastatic breast cancer? So this is Gabrielle Rock. She's a collaborator at University of Alabama. She went through a national database called SEER Medicare to find out how long people live with metastatic breast cancer. And as I told you about Peg, she lived over 30 years. But Gab will show you that about 99.8% of people die of metastatic breast cancer within 20 years. So it's a very unusual set of patients. So we began to ask, are there more patients like this here at the university? And we're able to identify about 53. They had different types of breast cancer. They had different stages at diagnosis. If you look at their history, the earliest ones were diagnosed in 1978, and some others were diagnosed in the 1990s and had lived for many years with breast cancer. And we began to ask, okay, is there something about the genes driving the growth of their cancer that's different than everyone else? Or is there something in their genes that they inherited from the parents, the germline, that makes them different? And so we began to ask that. So I got some, again, wonderful collaborators. Rachel Yan, who was an undergrad here, now a medical student in Rutgers, doing really well. Stephanie McGregor in pathology, and Jen Lafine. And we said, why don't we find these patients, get their tumors, and if they're willing to let us to do DNA tests, we can learn what's driving the growth of this cancer. And so we did that. We were able to find about 14 where we were able to get tumors. And we tested all the 20,000 genes encoded in their genome. And we found many different mutations, but we didn't identify a clear pattern. It wasn't crystal clear to say, this is why these people lived for a long time. So we thought, it's going to be challenging to answer this question with 14 people. There's such diversity of cancers. We need to have a larger group of people than we can find at one cancer center. So we decided to use social media, and this has been kind of fun because you start to use Twitter and things, and you say, hey, if you have metastatic breast cancer, would you like to help us understand how some people live for a long time? So we did that. Peg was in the newspaper. People started contacting me from all over Wisconsin. Then it started to go national, and we said, okay, let's just find a way that everyone can participate. So we got a grant. We created a web page here where our patients, people from anywhere can log on and click the Participate Now. They can give us all kinds of information about their breast cancer history. Over the past year, we've had over 700 people sign up from all across the country. We have every state covered except Wyoming. I don't know why not Wyoming, but we have everything else covered there. And we even had patients from international who signed up and said, we want to participate in this. So we got them to fill out, basically spend an hour filling out over 100 questions about themselves, but in partly to identify which samples that we could get from their local hospital to test their genes. I realized we had to visualize this data. And there's an emerging field called data science. So we ended up with these huge surveys on hundreds of patients. And so I said, what am I going to do with this? I want to see the data and use it. So, but I didn't want to do it. I didn't just want to create an Excel spreadsheet and do it once, and then 100 more people sign up. I have to do it again. So I was very lucky to recruit Hangjali Zhu and Grace Joyce. It's funny that you talked about the rowing event. Grace is on that varsity rowing team and is going to train nationally this summer. But when she's not rowing, she's working with Hangjali, and they're programming our scripts. And so they programmed these scripts that automatically take the latest data we have from our cohort who've signed up these surveys and generate these graphs. So this is the graph like I showed you from our small number of patients at UW, showing how long these patients' histories go. And so we have many breast cancer patients going back years and decades. And if you look from before 2000, we have over 100 patients who were diagnosed before 2000 with breast cancer and are alive today with metastatic breast cancer. So we're able to find these patients everywhere. And Grace and Hangjali kept doing more. They kept going and doing more and more. So this is information about where their tumors were in their body. And as you can see, it's hard to read this, but at far left is the patients who had metastatic cancer to bone. So that was the most common. But the metastatic cancer went to other sites. And then they began to examine what are the characteristics and habits of these patients. And so this is one example of one of the graphs they made, where we asked patients what their exercise was. Do you exercise more? Because many of the advocates around the country said, well, maybe people who exercise a lot live longer. So I'm not telling you not to exercise. That wasn't my message, but Hangeli and Grace, what they found was there was no correlation with the survival. That's not what drove survival, it looks like. So what we're doing now is we're picking these folks who are the longest survivors. And Madeline Sass, who's going to be a medical student here, and Kayla Lemon, who runs the Molecular Tumor Board, are doing a lot of work contacting these hundreds of patients across the US and internationally and trying to track down their tumor samples that are in some pathology bank in some hospital anywhere. So we're spending a lot of time talking to patients and asking their permission to do that. So that's all I want to tell you today. And so I just want to put this all in perspective of what I'm talking about. Just to give you a flavor of what's happening in the Cancer Center and how we're reaching out to the state. What the Molecular Tumor Board is trying to do is to help as many patients as possible across the state, using the technology we have today, using the drugs we're working on to help as many people as possible. And so our goal is to help and reach every patient with incurable cancer. We also want to do more. We want to look forward to the future and not just use the technology and the current knowledge. We want to learn from our patients. And PEG and these other long-term survivors with breast cancer is just one example of these unusual group of patients that we can learn a lot from. So what we hope to learn is how to make more patients like them, to live well with metastatic cancer if we can't get rid of it. So with that, I want to thank you, and I want to take any questions. I really appreciate your attention tonight. Yes?
So with this high throughput sequencing, a few years back they're saying, oh, one day it'll be $1,000 per patient, like less than an MRI. What's it at now?
It depends how you define that, but it's in that ballpark. It really is. The question was, how much does it cost to do DNA sequencing for a patient? What we do for our clinic patients is we're looking under in the genes that are most likely to help them. Yeah, specifically. Because the tumors have tumor cells and non-tumor cells, so we have a mixture of DNA in there. We have to do what's called a high depth. So we have to do a lot of sequencing of each gene we look at. So we're doing about a depth of 1,000. Meaning for any gene, any particular region of that gene, we want to see 1,000 sequences from that. So the real cost to Strata Oncology, who's running these tests for us, they estimate is about $100 a patient for the supplies, the sequencer. But they have overhead. That doesn't include, they have a company, and they have to, they have a building, and so, and they have a molecular pathologist interpreting all this. So it kind of depends how you do it. But for research, for the long-term survivors, it's $1,000 or $2,000 to look at all 20,000 genes in their tumors and in their somatic. So it's really close to what. Yeah, it's close. It's basically it's similar to an MRI. Yeah, yeah, yes.
You mentioned the particular drugs being developed and then decide which. How are drugs defined? I mean, if we're talking about a very small group of people with particular, what drives the generation of these drugs to begin with if the percentages are so low?
So the question was, what drives the development of a drug that is helping such a small fraction of patients? And the answer is, people are always looking for that next drug. And to have a really good drug, you can have a drug that works moderately well in almost everyone. That's a way to make money. It turns out, if you have a drug that works extremely well for a small fraction of patients, that's a boon too. We learned that with imatinib, or Gleevec, which was one of the first precision oncology drugs for the BCR-ABL gene back in 1999. And the company Novartis developed that. And at first they're like, why bother? There's only 2,000 patients a year in the whole world, or in the whole US, that have this disease. But it turned out it worked really well. And these patients who would have died or had a bone marrow transplant can be on these drugs for years. There's also an FDA process called an orphan drug process that some of these companies can use that gives a little more patent protection so they can earn the money back on developing it. But the companies that we're working with, one company, Loxo Oncology, was just sold to Lilly for an awful lot, developing these drugs for tiny fractions of patients. Because when those patients are found, they work profoundly well. Yes?
So out of 1,000 cases a year, how many individual patients can you actually help today?
So the question is, out of the 1,000 patients a year, how many are you going to help today? And I guess it depends on how you define help, and this is a problem in the field. For example, in lung cancer, doing this DNA testing has been standard of care for several years. So if we count those, we help more. If we don't count them, we help fewer. And it's also a moving target because, for example, any day now, we expect to have a new breast cancer drug that's applicable to 25% of breast cancers that have a PI3 kinase mutation. And we didn't have that drug, 2 months ago. So we're dealing with a moving target. So when we've looked at our data, we've recommended drugs for about 30% of our patients. But a smaller fraction of them actually get them for one reason or another. It can be it's been challenging to get those drugs. And if you define help, so if you define help by who got the drugs, it's probably closer to 10%. If you define it by who got the drugs and really did well, it's probably closer to down about 5%. And it's fun to talk about this with oncologists around the country and patients and advocates, because they have very different feelings about this. Some oncologists are very negative. They're like, you're only going to help 5% of patients. Why work so hard? But many of our patient advocates and my patients and I feel differently, that if you can help a small fraction of patients, it's worth doing. I don't know, you can give me your opinion, but we're not taking anything away, because they can always get lung cancer or colon cancer treatment if this doesn't work out.
Well, I think it's also, you're at the beginning, too.
We're also at the beginning.
It might be in 10 or 20 years, it could be very different, and it's only going to be with research at this stage.
Right, as we're bringing new drugs in, which we can only develop if we test everyone, as we're bringing new drugs in, the percentages are getting higher. Yes?
When you talk about percentage of people you can help, do you include the people who have, the people are going to find a clinical trial as opposed to a medication?
Yeah, so the question is, do you include clinical trial as helping the patient?
And what share of people get the clinical trial recommendation as opposed to a...
So since we've developed the program, we're bringing clinical trials. A larger fraction are getting a clinical trial recommendation because now we have them in Wisconsin. Like 3 years ago, what we saw is, oh, we found a clinical trial in Texas for you, but that wasn't very useful for our patients. So we really worked hard to bring them here through the processes we talked about. So again, this gets back to how you define it. We have a recommendation for about 1/3 of patients for a medication. About 10% of them actually get it, and only a fraction of them have benefit as defined as their tumor shrink or they're on the drug for a long time without evidence of tumor growth. And that 5% includes people on clinical trials, because some of these drugs in development are way better than the drugs we have today. And it includes patients who get, like the one who got the HER2 treatment. It includes patients like him who got a treatment for another type of cancer as not a research project. Yes, in the back.
With your patient-first paradigms, you still have to have the null hypothesis and double-blind studies, so some of your patients are not going to get the drug?
So the question was, do you have to have the null hypothesis in double-blind studies, and will some of the patients not get the drug? Many of the studies we're doing are called phase 2 trials. In oncology, most of our trials divide up to phase 1, where the drug is being tested for what dose and what side effects occur and what the pharmacology of the drug. Phase 2, where a large group of patients with a certain characteristic, like the genetic mutation, are treated, and they all get the drug. And phase 3, where it's randomized. Many of the drugs here in precision oncology have been approved after phase 2. There's been a lot of controversy about this. There was an article in the New York Times about 8 years ago now about melanoma patients. So what happened in melanoma is about a quarter of the patients have a mutation in a gene called BRAF. And there was a drug that worked really well and shrunk tumors in 70% of those patients in the phase 2 trial where everyone got the drug. And some people felt, gee, if I had that, I'd want the drug. It shrinks the tumors in 70% of the patients. And there were some people who felt that, no, we have to do the trial and show that we're doing a control group where we're showing that it makes people survive longer. And there was a lot of hand-wrenching. You can go back and find this article about this in the New York Times. What happened was they ran the phase 3 trial. It had a survival benefit, so people who got the drug did better than the people who didn't. And in the article, they actually told the stories of the people who were randomized to not get the drug. So it's a very hard thing. And I don't think there's very coherent way of dealing with that. I think some drugs like imatinib, Gleevec, and others are getting approved based on phase 2 evidence. They shrink tumors in enough patients, and the patients do way better than any patient in the past. We approve the drug. So there is some flexibility in that. One of the trials that we're running right now is a RET inhibitor, which has shown really great efficacy and better than all the FDA-approved drugs that are RET inhibitors with much less toxicity. So what they're doing is a phase 3 trial where patients are, 2/3 of the patients are going to get the new drug, 1/3 are going to get the old drug, and the people who get assigned to the old drug will have the option of getting the new drug after their tumor grows. So there are ways to deal with that, but it's not easy. And I don't think there's one answer, and the FDA has been flexible and willing to look at data without placebo-controlled randomized when that makes sense. Right here.
Do you have a plan to make this tumor board a national organization? That would seem to be logical for the next one.
So the question is, could we make this a national organization? What we've been asked over and over is, how are you going to do this for all the patients? Like, how are you going to make this scalable? And so what it's come down to is my partner and I looking at 50 to 90 reports every 2 weeks, picking out the 6 that are the hardest to decide, putting those for discussion with the group. And the others are things we've seen dozens of times, so the recommendations are pretty clear. There is a group called American Society of Clinical Oncology that tried to do this nationally. And frankly, it's been challenging to get that organized at a national level. There's such a diversity of what people do. The other thing is we have allowed people from other parts of the country to submit cases. We have a group in Georgia that occasionally submits us a case, but we can't bring the trials to Georgia. So there's some regionality that's inherently makes sense. You really want a central location where you can bring the patients in if they match a drug or a trial that you can bring there. Yes?
Can you give us a number for how many additional trials are now accruing people in Wisconsin that weren't before?
Oh, so the question is, how many additional trials have we brought and opened so far? I think we're, in the past year we've brought about 8. Yeah, and we're looking, there's more in the wings, but we're waiting for the patient to show up that needs it. Yes?
Do you see this being used on everyone who's diagnosed with cancer? I mean, being able to get the correct treatment for, right off the bat, rather than waiting until it's metastasized, would seem like
that would be a good thing. So the question is, do we see this used on every cancer patient? And the answer is yes, I think it will be in time. Once we have the list of effective medications for our metastatic patients, then that will then be integrated into clinical research with our patients who have potentially curable disease. The problem is we're not willing to take a risk that we're going to hurt, that our patients won't do as well. So for example, I treat patients with breast cancer. I see them every day. Some of them have stage 2 or 3 breast cancer. I give many of them chemotherapy, knowing that this works on the largest number of patients, and in the future, I might be able to do gene tests and find the right drugs for the right patients. But until I know I have the right set of drugs and they're very effective in incurable cancer, I don't want to risk not giving that patient the effective drug. So the way that many of these are being developed is some of these patients are getting the standard chemotherapy first, then they're getting surgery, and those with who seem to have a higher amount of cancer left behind can then be the ones to go on to these targeted drugs. So I think that is happening. It's happening now, but it's happening a little slower, and this is a little counterintuitive. Why wouldn't we want to use it on them first? But the answer is we don't want to risk losing lives of people where we have drugs with a really strong track record. And so we have to be very careful about how we bring that in.
Well, some of those drugs are amazingly toxic.
Some of those drugs are amazingly toxic. Yeah. Yeah. So I think this will supplant those drugs. The interesting thing is, you ask about how we go from metastatic disease to early stage cancer and be smarter about that, and I think that's happening slowly. When I started in this a couple of years ago, what was happening is every oncologist in the state was treating patients with metastatic disease with this standard chemotherapy, then this standard chemotherapy, then this standard chemotherapy, then would run out of the 3 standard chemotherapies and then run the testing and then send the test to us. And what we found from that is many of those patients were so ill, both from the treatment, but also that the disease had grown at the end of those 3 treatments, that they weren't in a position to get one of these targeted drugs. So we've been working just to get every patient right at the beginning of their metastatic cancer to get this test so we can plan out when they should get these targeted drugs. In the back.
Does the application of maybe a successful targeted drug open up traditional procedures later sometimes, or is it usually the end product, or it doesn't allow for some more traditional, let's say if a tumor shrunk, they could remove it, or a little easier, or?
So the question is, if we use the targeted approach, could we remove the tumor then potentially? So there's a very rare set of cases for the reason I'll explain in a minute. But the rare set of cases, for example, is there's that NTRK gene that I told you is very rare, but we have really great drug for. That's found in often children with cancer in these sarcomas, and sometimes those show up in the pelvis and they can't be removed because they're stuck to critical body structures. So there are a few cases where that drug was given for those patients with unresectable tumors where the tumor shrank and became resectable. And then the surgeon was able to remove the tumor. So yes, that can happen. In many of our patients, most of our adult patients, it's not really feasible because what's happened, the way their cancer became metastatic, is it started at the primary site, whether that's breast or colon and lung, went through the bloodstream, and these tumor cells scattered all throughout the body. So the problem is, my cancer center director likens this to dandelions. So once the dandelion, blows, you can find the first one on your lawn in the spring and rip it up, but you're not going to necessarily get rid of all the dandelions by doing that. So that's sort of the problem with most metastatic cancer. So really you need, to continue the analogy, weed killer. You need something that goes to all the areas of cancer everywhere and controls it for as long as possible. Yes?
You mentioned that you work with pharmaceutical companies as an incentive for them to give them pay for the test, so that then when they look at the patient, they come up with new drugs, precision drugs. Now the question is, do you think we should put more emphasis, instead of developing precision drugs, to encourage companies to come up with early detection of the disease, cancer? Like biotech companies should be support the universities more focused on early detection. And I think it's a trade-off. You want to treat after the metastatic cancer, or you want to put more money to study any early detection mechanism. And the second question I have is, do you have in your database the defective gene that is associated with different patients, so that their children and grandchildren, if they can access, then maybe they don't have to develop metastatic stage in case they get the cancer. You can immediately identify this is the defective gene that your parent or grandparent had, and we can quickly do the treatment.
Right. So the two questions are: how can we use DNA testing to find to detect cancer early, or know the genes in the family to prevent it? So, in terms of detecting cancer early, I think that's important, and I don't think testing DNA in tumors is mutually exclusive with prevention. I think they're both important. And so, for example, we're working with Exact Sciences in town here in Madison on some research projects where we're helping them devise DNA blood tests that can detect cancer very early. And so the idea is using that for screening. So yeah, there is an important role of early detection. I honestly don't think that's going to ever be enough to prevent cancer in everybody, because even when you detect it early, you have to do something with that. But the other part of the question was, can we use the information to help other family members? And keep in mind that what we're testing is the tumor. So a lot of the mutations we're seeing are somatic, meaning they're private to the tumor and they're not in the rest of the person. However, occasionally, as you bring up, the mutations we find in the tumor are not private to the tumor, they're in the whole individual. And so when we find a mutation that we think may be like that, we ask the patient, and one of the recommendations that we give in the tumor board is that they see a genetic counselor and get germline testing, usually blood DNA tests, to see if that's a gene that could be inherited in the family and to notify other family members about that risk for prevention. But I think that's very important, because I know today in oncology, because it's genetic counseling and testing is a little bit slow and it requires another patient visit, I think we're missing a lot of patients where we could find genes that are inherited in families and prevent a lot of cancer if we could do that more widely as well. Yes?
Tumors can have 2, 3, 4 different cell lines in them, if I understand correct. The first question, you said you do it 1,000 sequence. Is that how you deal with multiple lines, or is there a cytological approach first and then you do the DNA? I'm just wondering how you deal with the possibility of 3 or 4 lines and 3 or 4 different resistances to the drugs that you're trying to use.
So the question is, how do we deal with the polyclonality? It's the idea that, yes, the cancer started with one set of genes, but then made all these daughter cells which have different mutations in them that may do different things. And how do we know that, and how do we deal with that? Most of the genes we want to target with these drugs are there right at the beginning. So very rarely do we want to find a gene that's in only in a subset of the cancer cells, because that's not going to be a very effective therapy if it only kills or works on 10% of the cancer cells. So it is hard to do that. Most of these specimens are already dropped in formalin and in a paraffin block before we get them. What we can look at is what's called variate allele frequency, meaning out of those 1,000 DNA reads we have, what fraction of them have the mutation? And if we find, for example, this gene, it's found in 30% of those, so the variant allele fraction is 0.3. And this gene, it's only in 5%. That suggests to us that the second gene, that's 5%, may be only in a subclone, and it only may be relevant for a fraction of the tumor cells. So we do look for that subclonal mutations by assessing how many of those DNA sequences have the mutation. Now, most of what we're targeting and going for here are things right at the beginning. But a small number of these mutations emerge as a resistance mechanism. And that resistance doesn't really emerge in that specific way to chemotherapy, but to other targeted therapies, it does. And so it's really important when these patients get on the first treatment and their tumor shrinks, and then we see the tumors grow, to do repeat DNA testing to find out how those tumors became resistant. And many of those tumors have a mutation in a gene that really explains that well. It's a mutation that often causes the protein to fail to allow drug binding, for instance. And then you can get second generation and third generation drugs that overcome that in some cases.
So is it going to be analogous to, not the same thing, but will it be analogous to, people understand antibiotic resistance.
Mm-hmm.
Is this similar where you have something, you apply a drug, it kills most of them, but some of them remain behind, or there's a later mutation. So that's selection, and you're left with a resistant.
Yeah, so that's a very good analogy. The question was, is this analogous to antibiotic resistance, where you give an antibiotic and then only resistant bacteria grow back, and then you can't use that antibiotic? That's something that happens all the time in oncology. And it's a problem even with these targeted drugs. And for some reasons, some of them it takes years to develop resistance, and other ones people can develop resistance in a matter of months. So there's a lot of variability in that.
Any other questions?
Well, thank you all. I appreciate it. And